on no, wait. On Tuesday, we talked about mutations, right? We talked about how chemicals cause the mutations, either by inserting themselves instead of the normal nucleotide or getting in between the base pairs, how physical factors cause them, UV light forming the thymine dimers, or gamma and X-ray radiation that breaks the chains. But we never actually got to the point to discuss how these mutations can be fixed. So we have discussed some points here, and DNA polymerase performs the actual proofreading, okay, and replaces incorrect bases that do not match bases that were inserted and do not form complementary pair, right? Now, what if it's found later? Again, if you think about the double strand, you have complementary pairs, and if this is not complementary, you have a distortion in a double helix. That distortion in the double helix will be recognized. The fragment here will be excised, and DNA polymerase 3, the same enzyme that made the new strand in the first place, will build the correct one. Okay. Now, one example of repair that I wanted to show you is the repair of thymine dimers, because those occur most frequently. We are not exposed to acridine orange or ethidium bromide on a daily basis. And if you don't smoke or you don't inhale from exhaust pipe of your car, then you're not exposed to polycyclic hydrocarbons as well. So UV light and X-ray and gamma rays are not frequent, but UV light is always there, pretty much always, especially if you live in Florida. So how do we fix the thymine dimers that appear in the cells? There are two ways. One that occurs in the dark, and another one that occurs in the light. So repair in the dark is fairly straightforward. We kind of discussed it already. The thymine dimer, illustrated right here, okay, is excised. And it's interesting what excises it. It's a helicase, the same enzymes that unwinds the DNA. As you can see, one enzyme may have several functions. Does that make sense? So it separates the strands further and cuts the piece of the affected strand out. That makes sense? And then DNA polymerase 1, the same enzyme that replaces RNA primers during the replication, okay, the same enzyme fills up the gap. Now if you think about it, this is your DNA after the excision, okay. When you fill it up, you will still have to connect these loose ends. That's the job for DNA ligase. Remember the enzyme that connects the Okazaki fragments on the lagging strand. So DNA polymerase 1 will fill the gap, and DNA ligase will stitch the newly synthesized fragment back into the strand. Another ways you can imagine, if this was in the dark, then another one should be in the light. This is called photoactivation obviously, quite frequently we get those mutations, the thymine dimers, when we are exposed to the sunlight, and we can see that sunlight. UV light is an invisible component of this. When UV light as a component of the sunlight causes the formation of thymine dimers, the visible component of the spectrum activates the enzyme that is called photolyase. And photolyase does not cut the fragment out. Instead, it breaks the bond between the thymine dimers and the original pairing is restored. Does that make sense? You see the difference? So one, it's kind of one is let's destroy what's broken and build it from scratch. Okay, that's the excision repair. And another one is let's not destroy anything. Let's just nicely and accurately break the bond between two thymine dimers, so the pairing will be restored. Pairing. Now look at this again. Let me erase it. So we start with a problem that UV light right here causes the formation of the thymine dimers when two adjacent thymine residues in the same strand are connected. When they are connected, the normal hydrogen bonds between the thymine and the adenine on the other strand cannot be formed. Does that make sense? Say, if I know, like if say Rachel and Jamie are supposed to hold hands, and Mike and Kashida are supposed to hold hands, right? That's the normal pairing. But if Mike and Rachel would hold hands, they won't be able to do it across the table. Does that make sense? So those bonds here, that bond is a covalent bond, and it prevents both thymines from establishing hydrogen bonds with these two adenines. So what photolyase does, it breaks down that covalent bond, and the pairing, normal pairing, is restored. Does that make sense? Okay. So mutagens, how do we know if something is mutagenic? For this we use so-called mutagen test. And it's also called Ames test, and it's kind of perverted in a way it's done. So mutagens, mutagens sort of randomly change sequence of DNA, right? The bacteria are extremely convenient for the mutagen identification. Because of what? What do you think? Why bacteria are convenient for identification of mutants? Why they are more convenient than, let's say, mice? Oh yeah, they fast, right? Much cheaper, and they standard, yes. So standard, faster, cheaper, and having 1 million bacterial cells isn't a problem. Try to imagine 1 million mice. I cannot. My personal record in my personal colony, well, not personal, but 96, and it was a pain in the ass to handle all of them. Okay? So 1 million mice, I don't even want to think about it. So fast, cheap, standardized, easy to manipulate, and there are a lot of them. So there is a higher chance that in a given time you're going to get mutation introduced in one of the genomes. So let me start with the first part of the Ames test. The organism that is used in the Ames test should have mutation itself. The organism that is used in Ames test is auxotrophic bacteria. Can anyone tell me what auxotroph means? We talked about it. Auxotroph. Bless you, it requires something. Auxotrophic microorganisms require something. Say again? Okay, any nutrients? Specific. They need some specific nutrient. They cannot make it. So what is done? You have a colony, many colonies of Salmonella. Okay, they grow on the medium, and then you do the replica plating. So you have this, I don't even know how to call it, the transfer pad, okay? And you attach the transfer pad to the initial set of colonies and move that pad to two different media. One media that has histidine and another one that does not have histidine. It may be histidine, it may be some other amino acid. It doesn't really matter that much. Okay? Make sense so far? So then you look at the colonies. Obviously, that media that has all amino acids, including histidine, will support growth of all kinds of bacteria. Well, I mean, it's all Salmonella, okay? But if you would look at the plate which does not have histidine, you may notice, if you compare them, you may notice that some colonies are going to be missing. So those missing colonies on the histidine negative plate, that colony will be missing on the histidine negative plate. So that colony is auxotrophic microbe that can grow only when histidine is present in the medium. Does that make sense? Okay? Does that idea make sense? I'm going to pause here and make sure that, okay, so it's an exclusion, okay? So we find the microbe that requires histidine. So it has mutation in its DNA already. It's a random mutation. It's accidental mutation. It is not caused by anything. Does that make sense? It was there maybe in the very, very beginning, you know, because there are some varieties in the genome. That make sense? No? I want to make sure that it makes sense. Okay, so we identified that microbe that requires histidine. What do we do next? So we take auxotrophic Salmonella typhimurium that we just isolated here, okay, and we expose it to the mutagen, right? And then we plate it. On the medium that does not contain histidine. Does that make sense? Now, only colonies that can make histidine will grow. Does that make sense? Now, if mutagen actually causes mutations, then there is a likelihood that microbes that cannot make histidine will revert to the normal phenotype, to the phenotype when they can make histidine. and the number of colonies on a histidine negative medium will increase. That makes sense? Let me do something that I did before. Okay, I'm gonna draw. Does that make sense? ST is Salmonella typhimurium. Okay? And it's histidine negative. So we have two Salmonella, two different setups of experiment with the same microbe. So before we proceed, what does that mean, histidine negative Salmonella typhimurium? It cannot make what? So what has to be present in the medium? Histidine. Very good. We plate it. On the medium that does not contain histidine. Is this microbe going to grow? No. If we have a colony, what does that mean? Say again? Yes. So it is these microbes, the colony, are not histidine negative anymore. They have reverted to the original phenotype. Does that make sense? Reverted. Okay? Can that happen spontaneously? Sure. Make sense? So far, you follow me. Okay? You see, we didn't add anything, we just plated. That's a control. Okay? Now, this guy will be plated on the histidine negative medium, but here we have mutagen. Good? You follow. Are you following me with this? Yes. Say again? No. Mutagen is some sort of a chemical. You want to test some kind of a chemical. I don't know. A component of the dishwasher fluid to see if it's carcinogenic or not. Okay, so you treat microbe with that mutagen, and then you plate it on the histidine negative medium. Now, if it's not mutagenic, the chemical, are you going to get any revertance? If it's not mutagenic, no. Bacteria is not going to go back to original phenotype. But if it's mutagenic, that histidine negative microbe is going to come back to the original phenotype. Does that make sense? Those are Does that make sense? Does that make better sense now? Huh? Say again? Sure, and it's all on video. Yeah, it's a screencast. It's on the YouTube. Gonna be on the YouTube. Okay. So, we good? Now, let's go back here. So since now it makes more sense, I don't hear one question from you. Why the hell do we need the liver extract? Well, it kind of is explained there, but in a very short statement. Liver, it not only takes good, it excretes toxins, but another function of the liver is conversion of chemicals. And sometimes a chemical, the mutagenic chemical, can be either neutralized or actually can become stronger if liver enzymes somehow change it. Does that make sense? I am no pharmacologist, and I won't be able to give you the exact pathway. Have you heard about the drug called metronidazole? It's a very, very broad spectrum antibiotic that treats anaerobic organisms. Anaerobic fungi, anaerobic protozoa. Well, for fungi, clotrimazole and fluconazole are more common. Metronidazole, all kinds of anaerobics like Clostridium difficile, all kinds of anaerobic protozoans like Trichomonas vaginalis. So, but the funny thing is, well, not so funny, but metronidazole is a precursor. When it gets into the organism, into the body, it is converted into the actual compound that destroys the anaerobes. So it's not an active molecule. It is converted. All right? And liver can do that. So that's why liver extract is added to control for this. That makes sense? Now, does that mean that if it's a mutagen, we just say, oh, that's a mutagen, let's kick it out of all the chemicals? Of course not. You can test it further. But now you know that there's potential mutagenic activity. You can test it, say, in mice. Okay, you can see if it causes, I don't know, cancer or something else horrible. Does that make sense? Now we're going to talk about sex. Well, just a little bit. The bacterial one. Not only this. So, the purpose of sexual reproduction, as we have discussed, is to increase genetic diversity. But bacteria do not have sexual reproduction. But as we can see, they have amazing genetic diversity. We just saw that example with Salmonella, which can become auxotrophic strain and then revert back to normal. Right. Other implications of genetic diversity we can find in the hospital in the form of what? Bacterial genetic diversity. Yeah, antibiotic resistance. MRSA, yeah, and I wanted to kind of discuss it with you. But first we have to understand how diversity operates, how it can be maintained. Now in humans, when people or mammals or any organisms that have sexual reproduction, bless you, when they produce progeny, they exchange genetic information. Do you agree with me? That's what happens. If we would clone humans, clones would not have the genetic diversity that the products of sexual reproduction would have. So bacteria have to exchange the DNA somehow. When bacterial cell divides, the DNA of two daughter cells, how they are related to the DNA of the parent cell. Are they same? Are they totally different? Generally the same. Yeah, maybe few mutations, but generally they are the same. Okay. So the diversity in microbial populations, and especially in bacterial and archaeal population, is maintained by the various mechanisms of the exchange of DNA, exchange between the cells. Okay, it's called often horizontal gene transfer. Horizontal gene transfer, and it's different from vertical gene transfer. when organisms with sexual reproduction produce progeny, they have vertical gene transfer. Make sense? Bacteria, they do transfer genes, but it's the same genes. There's no, really diversity here. So there are three basic mechanisms of horizontal gene transfer. Transformation, when DNA, either in the form of the plasmid or in the form of a fragment of bacterial chromosome, is taken by the bacterial cell and is either incorporated in the genome or, alternatively, it's not just shown in this picture, may exist, remain a plasmid in the cytoplasm. Make sense so far? Another way is transduction. We haven't discussed the viruses yet. And that's one of the, not the problems, like little obstacles that we have to overcome during this course. Different parts of different aspects of microbiology, different areas, are so connected that you cannot talk about bacteria without mentioning viruses of bacteria. You can see those fancy models right there, right behind Julia. Those glass models. That's the model of bacteriophage infection. So bacteriophage is a virus, generally speaking. Remember, an acellular structure that can infect bacteria. Okay? So if during the break, if you want to wonder about how it happens, it inserts its genome, its DNA or RNA genome inside of the bacterial cell. Proteins of the bacteriophage are synthesized by the bacterial ribosomes and stuff. Phage self-assembles, packs its DNA into the capsid. You see those like alien spaceships? You see the alien spaceship structure right here? It packs its DNA and leaves the cell, and bacterial cell explodes. During this processes, sometimes phage DNA becomes incorporated into the bacterial cell, or alternatively, phages can actually transfer fragments of bacterial genome from one cell to another. I will illustrate it for you. And finally, conjugation. This process is sometimes, in the jargon, referred to as bacterial sex. By the sex pili, bacteria exchange the DNA. Okay, we're going to talk about each of these processes in more details. And the first is going to be transformation. It was an experiment of Frederick Griffith, I think like 40s, something like that. He was studying two strains of Streptococcus pneumonia. The pathogenic and the non-pathogenic strain. Pathogenic strain was called S strain, smooth, because it had capsule. The smooth is kind of exemplified here. Okay. The non-pathogenic strain, or rough strain, did not have capsule and is shown here. Okay. So his experiment was, he took non-pathogenic strain, inoculated bacteria into the mouse, and the mouse survived, which is not really surprising. Then he took pathogenic strain, but you like mice, these guys? I really like the pictures. They're really cute here. Huh? They're really, really cute. Then he took a smooth strain, the pathogenic one, and destroyed it with a hit. Killed bacteria with hit. And inoculated that inactivated bacteria into the mice again. And kind of expectedly, mice survived. by the way, before those experiments, he demonstrated that just if you take just a smooth strain, the pathogenic one, and stick it into the mice, mice die. Does that make sense? So kind of one thing is missing. So we can put smooth mice death. Okay? So when you inactivate pathogenic strain, mice survive. But then he did an interesting experiment. He took a live rough strain, which is non-pathogenic, and inactivated smooth strain, which, when inactivated, is also non-pathogenic, right? So two non-pathogenic samples of microbes, one live, one dead, inoculated them together in the mice. The mice died. So what he did, he isolated the microbe that killed mice and showed that this is the smooth strain. So apparently what happens, when you take a rough strain, which is non-pathogenic, and you add smooth strain, which was killed, that's why I put it in green, it produces smooth strain that is pathogenic. For a pretty long time, the idea was that it's proteins that are transferred from one strain to another that confer pathogenicity. But later it was demonstrated it's a DNA that confers that pathogenicity. So what happens? When you have the heat-killed smooth strain, free DNA exists there. By itself, it does not kill anything, right? But when you mix that pretty much free DNA and the non-pathogenic live bacteria, that free DNA from smooth strain gets taken into the cells that were non-pathogenic. Does that make sense? Like in an alien. So that free DNA transforms the non-pathogenic strain into pathogenic. Does that make sense? So during transformation, bacterial cells take in the free-floating DNA of other bacterial cells. And that is widely used in the lab, actually. Not so much with the fragments of the chromosome, but more with the plasmids. So this is an example of the plasmid. Circular, remember we talked about extrachromosomal DNA and we talked about a plasmid. So plasmids are DNA fragments that are relatively easy to operate in vitro, in a tube, in a test tube. You can modify the genes that are present in the plasmid. And this is some generic example. Imagine that you have some protein that you want to make. Say you want to make insulin. You take insulin gene and using fairly straightforward methods, insert that gene into the plasmid. Right? You have free plasmid, free DNA. And then you mix plasmid and some bacteria that you can grow on the agar plates. In the lab, it is usually E. coli. And you induce heat shock. You expose them to 42 degrees Celsius, not so much hot, not very hot, but enough for them. So bacteria take in the plasmid that is in the solution. Does that make sense? That's just what you do in the lab. And then they start to produce insulin, because that's what they do, because that you have a plasmid here that contains the gene. This is the promoter that will drive the expression of that gene, and they're going to make insulin. The only problem is that some bacteria will take up the plasmid, some bacteria will not. How can you separate them? Your plasmid should also contain gene that confers resistance to antibiotics, because then you can grow your microbes on the plate, the agar plate that has antibiotic in it. That makes sense? Only the ones that have plasmid and are resistant to antibiotic will grow, and you can select. It's absolutely standard method in the lab. It's regular one. Okay? Does that make sense? Now, this poses the problem. Again, imagine how easy microbes can exchange the DNA. Even if microbes, and that DNA does not have to be from the same species. If some species of the microbes die, cells die, DNA is free floating. Other bacteria can take this DNA in. It may be beneficial, it may be not. But imagine how many cells there are. Millions and millions of bacterial cells. And transformation will allow them to exchange the genetic information all the time. Does that make sense? Transduction. So, let me do this. Let me think how to do it better. Okay, I'm gonna do the separate slide again, if you don't mind, and explain the generalized transduction for you. Okay, I'm gonna do it very simplified way. So this is gonna be a bacterial cell. We're gonna try and make a little movie out of it. Okay, bacterial cell, and this is gonna be its circular DNA. Follow? This green guy is going to be a little bacteriophage with, bacteriophage with its DNA in it. Okay, that makes sense? I'm not really good artist, so I'm not drawing those alien spaceships. Okay, so I told you before, when phage infects the bacteria, it only inserts its DNA. So we've got DNA here. Are you with me? Okay. So what happens next? If you are a microbial cell and you have DNA, what you going to do with it? Can you express it? Can you express the genes? Why? It's the DNA. You can. I mean, stick in plasmid, you can express genes from the plasmid. That's what bacteriophages use. They have DNA that bacteria, they exploit bacteria, okay? They use bacterial expression system. They use RNA polymerase and ribosomes to make their own proteins. Does that make sense? They sneak in, they're parasites, okay? So phage DNA produce mRNA, and then phage proteins. We good so far? Now, proteins, what are they going to form? They're going to form phage particle, right? They're going to. That's actually an interesting feature of viruses. They self-assemble. They just like Lego. But Legos are not self-assembling. These guys are. So you're going to get those new phage particles, right? Now imagine what's going to happen to the cell when, and there are hundreds of phages, in one bacterial cell, what's going to happen to the bacterial cell in the end? What do you think? It's going to die, right? It's going to explode. What's going to happen to bacterial DNA? It's going to break, right? So here's what happens. Sorry. When DNA breaks in pieces. Okay? Now, when there is such a crowd of phages, when there are so many phage particles are produced, some of them will accidentally package bacterial DNA sorry, instead of the phage DNA. Does that make sense? Does that make sense? The final step would be for this cell to get totally destroyed. Okay? But these guys will go to infect other cells. Does that make sense? Now, this bacterial DNA fragment will get transferred to another cell. Does that make sense? You want to go over it one more time? Yes, we'll get to the phage one, okay? Yeah, bacterial DNA can get transferred by phages. So they accidentally package fragments of bacterial DNA when they leave the cell. Does that make sense? And they carry those fragments to other bacterial cells. And they may carry it over. That's another way for the microbes to develop resistance, to get those genes transferred from one cell to another. This is called generalized transduction. Okay? Now, I'm kind of giving you a heads up. So this cycle, when bacteriophage enters the bacteria and blows it up during the replication, is called lytic cycle. It makes sense that it's lytic, correct? But in my opinion, much more interesting. So that's what we were talking about. Those 2 bullet points talk about the slide that we just pictured. Okay, again, it's all on video, so you will be able to review it. Now, there are other types of phages, so-called lysogenic phages. Have you heard about herpes virus? Well, I believe you have. Have you? What's so interesting about herpes? What's the difference between herpes virus infection and, say, influenza infection? Ooh, okay, wait. So when, yeah, herpes can become latent, and it will become latent, which means once you get infected, it's with you forever. Okay, it's gonna get in your neurons and gonna stay there till you die. Okay. Well, yeah, love comes and goes. That's true. Yeah, and herpes is better than love. That's the gift that keeps on giving. Okay. Probably best Valentine's Day gift that you can give to another person. It's herpes. Yeah, it stays latent. When it gets reactivated. When you get cold sores. Why do we call them cold sores? Why do we call the herpes lesions cold sores? Yes, because when you have cold, your immunity goes down and herpes reactivates. right? But other than that, if you're healthy, it's not going to kill you ever. You're going to be fine, okay? So, influenza, if it hits you hard. You either don't have any symptoms at all, that can happen, or you get sick. There's no latency involved. It doesn't get reactivated, thank God. Okay? So, lysogenic phages are like bacterial herpes. They get in the cell, well, not the entire phage, but DNA of the phage gets in the cell and inserts itself into the bacterial chromosome. It integrates. Does that make sense? And then it sits there. Okay? At some point, they're under different conditions. This phage DNA gets excised from the chromosome, okay, and starts to produce phage proteins. Phage proteins organize in the get organized in the particles. Particles package the DNA in. You can see little DNA molecules in, okay, and those guys get out of the cell. Well, here's the trick. Excision is not always perfect. So in some cases, this phage DNA will also carry a little fragment of the bacterial DNA. Okay, so it's not cut exactly. There are some leftovers of bacterial DNA on the sides. And that bacterial DNA will be packaged into the phage fragments as well. Does that make sense? And will be transferred to a new cell. Now this phage, the one that introduces its DNA into the bacterial cell and DNA gets inserted into the genome, is called lysogenic. Okay? Which means it can produce lysis, but will not immediately produce lysis, unlike lytic phage. Lytic will kill the cell. Lysogenic will probably kill it in the long-term perspective. The question that arises right there is, all this, should it happen? Always. The answer is no. In some cases, the phage DNA gets incorporated into the bacterial DNA forever, and phage never gets reactivated. Does that make sense? No? So, phage introduces its DNA into the bacterial chromosome. And that's it. It never becomes the phage again, that DNA. Just some reason that happens. Why I talk so much about transduction? We heard about cholera. Cholera produces cholera toxin that messes up with the chloride transport in the intestine. So chloride, instead of being taken into the epithelial cells of the intestine, stays in the lumen of the intestine. What happens next? It's cholera, people. Diarrhea. Not just diarrhea. All capital letters diarrhea. Every 20 minutes diarrhea. 20 liters a day diarrhea. So yeah, the treatment is hydration. But the point is that toxin is encoded by phage. Ages ago, I don't know how many years ago, or hundreds of millions of years, that phage incorporated itself into the genome of Vibrio cholerae. It never gets reactivated anymore. It stays there. The funny thing, if you manually, sort of artificially excise that gene, that one phage gene, and you can find it in other phages, similar genes. If you excise that gene, Vibrio cholerae will be perfectly fine. It will not cause diarrhea. Microbe will replicate and everything is going to be okay. It's going to grow, but it's not going to cause diarrhea. Ever heard about diphtheria? Same goes. Diphtheria toxin is also a product of phage. You can excise that gene and Corynebacterium diphtheria will be absolutely fine. So phages contribute to the pathogenicity a lot. Well, spore is, endospores can be considered a virulence factor, but C. diff also produces toxins that destroy the epithelial lining. The cholera toxin does not destroy the lining per se. It blocks the transport of chloride. So chloride stays in the lumen instead of going into the blood. And because of the osmosis, water starts to rush into the lumen of the intestine and you flushed. Okay? Toxins have a variety of effects. So this transduction, when phage, lysogenic phage, carries over fragments of the bacterial DNA, is called specialized. Okay? That's a specialized transduction. Okay? So phage walks in, incorporates its DNA, well, not phage. Phage DNA is incorporated in the bacterial chromosome. At some point during reactivation, phage DNA is excised, but with a piece of bacterial DNA, and that piece of bacterial DNA can then get transferred into the new bacterial cell. Okay? Conjugation. Now, transformation, as you can see, the very first topic that we covered, is somewhat non-specific, right? Just taken in DNA. Transduction, whether generalized or specialized, is more specific, and phages contribute mostly to the virulence factors. Okay, they often represent virulence factors. Now, conjugation is a critical mechanism of antibiotic resistance spread in the populations of microbes. So during conjugation, you have two cells, fertility positive, the donor cell, and fertility negative. F stands for fertility, and actually it's fertility plasmid. Now, it doesn't mean, that means that this plasmid has a capacity to transfer itself from F positive to F negative cell. Does that make sense? So when F positive and F negative cell encounter each other, they form bridge using pili. The plasmid replicates itself and introduces the copy here into the fertility negative cell. Okay? That makes sense? So now you have two fertility positive cells. So what's going to happen if you introduce a microbe with a fertility positive plasmid in the population? You're going to have a spread of that plasmid through the entire population, and if the plasmid carries antibiotic resistance, the entire population will be resistant. Michael mentioned MRSA. What it stands for? Methicillin-resistant Staphylococcus aureus. Staph aureus is what it affects, you know. Staph aureus causes skin and wound infections, can cause respiratory infections, but we're concerned about MRSA because it's an important nosocomial pathogen. Nosocomial means, let me write this word for you. Nosocomial, okay, MRSA, MRSA. So nosocomial means transmitted in the hospital. To give you a perspective of importance of nosocomial infections, do you know how many people die every day of nosocomial infections in the United States? Huh? No, not thousands. But the example I really like, do you know the jumbo jet? It accommodates about 270 people. Imagine that every day one jumbo jet goes down and all people die. So that's how many people die every day in the United States because of the nosocomial infections. That's, yeah. The what? Technically, yes. Of course, it can be everywhere where people with MRSA can be admitted. What I mean is, so think about this. What happens is you start treatment of a microbe with methicillin, which is advanced form of penicillin. Okay? It inhibits the synthesis of bacterial cell. Well, we'll get to the mechanisms. Okay? If you're not following through with the regimen, if you don't give sufficient dose, and that's going to happen, microbes, you put selective pressure on the microbial population, so microbes will eventually develop resistance. Only ones that have intrinsic resistance or the ones that acquired somehow will survive. And if that resistance gene is on the plasmid, and if it's a fertility plasmid, that trait, the resistance, will spread through the population. Now you have MRSA that can be everywhere. Maybe your doctor that admits you, your family doctor, also practices once a week in a hospital. Your doctor can bring MRSA into the office inadvertently. And one of the things now is that perhaps MRSA is the new reality. It's a new normal. We should be careful about it, but we treat it as, oh my God, it's MRSA. Well, we have this microbe out there. Let's be aware of it. We cannot do anything about it now because it's spread so much. We can't limit this. Okay. Now, another way for spreading the conjugation works is formation of so-called HFR, high frequency recombination cells. So if fertility plasmid incorporates itself into the bacterial chromosome, it forms so-called HFR cell, right? High frequency recombination cell. Now, what can happen next? Because of the bacterial recombination process, which I'm not going to go into molecular details, the idea is that in some cases, the plasmid that was incorporated can get excised from the chromosome back. And as you remember from the example with the specialized transduction, excision is imprecise, pretty much always. Does that make sense? It's not excised to the right points. So, you have now a plasmid which carries with it a little fragment of the bacterial chromosome. This plasmid is still fertility plasmid, but it's not the same fertility plasmid as here. That's why we call it F' plasmid. Okay? It incorporates in the chromosome, it is excised from the chromosome, and after the excision, it carries over with it a little bit of the bacterial chromosome. Okay, it's like somebody stays at the hotel and, when leaves the hotel, picks up a couple of towels. Something like that. That makes sense? So that also can contribute to the transfer of actually chromosomal fragments from one bacterial cell to another. Another interesting thing that incorporated fertility plasmid can do, it can try to transfer the entire chromosome into the new cell. Now, how big plasmids are? it is varied, but they're usually rather small, 5,000-10,000 nucleotides. Chromosome is 5 million. It's much bigger, 1,000 times bigger. Okay, so when that F plasmid tries to transfer the chromosome through the pilus, this process takes a lot of time. We talk about minutes. And usually the pilus here is broken before the entire chromosome is transferred. Does that make sense? Yeah. So essentially, this cell, the cell that the recipient cell will receive a piece of the original chromosome. It is used for so-called genome mapping. Okay, so you can actually see how much time it was, and then you can see which genes were transferred. Okay, so you see, this is how much time it takes to transfer, I don't know, PABB gene. This is how much time it took to transfer PYRG gene, whatever gene it is. Okay, so you can say, okay, this gene is located kind of closer to the origin of transfer than this gene. It's interesting that the chromosomal map is mapped in minutes. For the same species? Yes. Yes. So if we talk about the same species, it's going to be standard time. So you can say that for all E. coli of a particular strain, it's going to be like this. For Salmonella, it's going to be like this. Okay? It's kind of a neat routine technique that will probably become obsolete in several years, since we now have such amazing sequencing machines. Okay, before we get to the regulation of the transcription and stuff, let's take a break, and then we'll chat about this.